Hello everyone and welcome to ITLS Academy Empower the Youth. So today in this lecture we will going to study about detection of endotoxins in food sample by Limulus Amoebocyte Lysate test. So in this particular lecture we will going to study about how you detect endotoxins in food sample. So before understanding this whole experiment, let us understand what does endotoxins mean. So endotoxin, it is a type of a pyrogen. Pyrogen means a natural compound that is found in the outer cell membrane of gram-negative bacteria, and they can impact over 30 biological activity. So endotoxin can lead to cell death by initiating the complement activation. So if you see this endotoxin, so it is a type of a natural compound, and this natural compound, it is present in the outer layer of the gram negative bacteria, and since it is present in gram negative bacteria, so they can impart, means they can do a you can say biological activity up to 30 biological activity. Okay, so it leads to a death of the cell, it leads to the sudden death of the cell by you can say by initiating the complement activation. The Limulus Amoebocyte Lysate, that is LAL test, was commercially introduced in 1970s. So this LAL is derived from the blood cells or ambiocytes of the horseshoe crab Limulus polyhemus. So it was firstly introduced commercially in 1970s, and it is also known as LAL test, that is Limulus Amoebocyte Lysate test, and it is derived from the word that is blood cell, or you can say ambiocytes of a horseshoe crab, that is Limulus polyhemus. Now let's move to the next, that is who is the scientist behind this discovery. So Frederick Bank and Jack Levin discovered that blood cells from the horseshoe crab were found to clot in the presence of endotoxin, and this technology was used in the development of endotoxin detection assay. So who was the first two scientists? So the first two scientists was Frederick Bank and Jack Levin, who firstly observed this whole thing, that a blood cell from the horseshoe crab, so they are capable, or you can say they were found to clot in the presence of endotoxin. So by using this technology, it was further used in the development of endotoxin detection assay. So Limulus Amoebocyte Lysate test is an aqueous extract of blood cell, that is ambiocyte, which is obtained from a horseshoe crab, that is known as Limulus polyhemus. LAL reagent react with the bacterial endotoxins, or you can say lipopolysaccharide, that is LPS. So all these full form you should always be remembering, that what do you understand by LPS, that is lipopolysaccharide. What is LAS? So LAS, again you should always remember that what do you understand by this LAL. So LAL is the L for luminous, and A for ambiocyte, L for lysate test. So you should always remember all these three thing, and it is firstly used by which two scientists? So the first two scientists was Frederick Bank and Jack. So now let's move further and let's see what is a luminous amoebocyte lysate test. So if we talk about this test and how this test is used in the detection of endotoxin in food sample, so let us understand that what this test is basically. So if you see this, we live in a very advanced society, and there is a very high demand of high quality of food. Okay, because nowadays people are becoming so much aware that they want to consume those particular amount, those food items that are contaminant free, or you can say those item that is free from any source of microbial attack, or you can say contamination. So so to test, so LAL test is used. So now let's move to the aim of the experiment, that is detection of endotoxin in food sample by Limulus Amoebocyte Lysate test. So what is the theory of this? So the microbial toxins are organic poisons produced by microorganisms. When such poisons are ingested, absorbed, or otherwise introduced into the body of different living organisms, they can cause damage to the tissue or to interfere with normal physiological functions. So we all are familiar with this first point, that this microbial toxins. So what are they? They are so much toxin, or you can say they are organic poisons. They are basically produced by microorganisms, and if such poisons are injected, or you can say if they are absorbed inside the body of living organism like human, so they can cause damage to the tissue, as well as what they do, they interfere the normal physiological functions. So microbial toxins, they are very complex in terms of structure and chemical composition, and they possess are antigenic properties. So that is the reason microbial toxins, they are a very complex term of structure and chemical composition, and they have so much they can cause poison up to an extent that they can cause the damage to the tissue, as well as they can cause a physiological changes in the human body. So these microbial toxins are very harmful. So it is very important to avoid this microbial toxin to get introduced into the human body. So there are very numerous type of microbial toxins that have been described. So these are bacterial toxins, fungal toxins, legal toxins, etc. So depending upon the type, means if they if a there are different types of toxins that are there, means some are bacterial toxins, some are fungal toxins, that is also known as mycotoxins. So we have already discussed about the mycotoxin in our cereal and bakery experiment, that what are mycotoxins, how they are produced. We have also learned about the different types of mycotoxins, and there are some legal mycotoxins, that is phytotoxin, etc. So exotoxins are up now. There is a change, means there are two types of toxins. First is endotoxin and other is a exotoxins. So what are exotoxins? So exotoxins are soluble substances that are produced in cell but secreted to the cell exterior environment. Okay, how they are secreted? To the exterior environment during the period of the active growth. So endotoxins, they are bound to the bacterial cell wall, means exo means outside, so they are secreted to the cell's exterior environment. Okay, but whereas endotoxins are concerned, they are bound to the bacterial cell wall as a structure component, or in, you can say, in certain types of microbes containing within the cell cytoplasm. So now let's talk about the flow chart, but let us talk about the differences between the endotoxins and exotoxins, because sometimes in interviews also they can ask you what is the difference between exotoxins and endotoxins. So you must be familiar with the difference between an exotoxins and endotoxins. So if we talk about endotoxins, so endotoxins are LPS, means they are located at the outer membrane, means outer membrane of gram negative bacteria, whereas exotoxins, they are polypeptides, proteins, but they are located on the extra chromosomal genes. Endotoxins, they are secreted when the bacteria die. Okay, after a bacteria get dead, or you can say जब bacteria मर जाता है, then endotoxins are secreted. But if you talk about the exotoxins, so they are secreted by the species of bacteria. Okay, so basically this is the basic difference between the exotoxins and endotoxins. So again, if you want, I can can write the question. What is the, you can just search on the Google, or what all the differences I have told, you can just see that what is the difference between exotoxin and endotoxin. Okay, so again I am telling you, you can note it down somewhere. I'll be speaking very in बहुत मैं धीरे बोलूँगी. So exotoxins are protein that are produced inside pathogenic bacteria, most commonly gram-positive bacteria, as a part of their growth and metabolism. Again I am repeating, exotoxins, they are proteins produced inside pathogenic bacteria, most commonly gram-positive bacteria, as a part of their growth and metabolism. Whereas endotoxins are lipid portion of polyliposaccharide, that is LPS. LPS is a lipopolysaccharides that are the part of the outer membrane of the cell wall of gram-negative bacteria. Again I am repeating, they are the lipid portion of lipopolysaccharides that are the part of the outer membrane of the cell wall of gram-negative bacteria. So this is the basic difference between endotoxins and exotoxins. Now let's move further and let's see the flow chart of LAL. So this is the flow chart of LAL test mechanism. So you can see this flow chart, how this endotoxins, that is LPS, is there. So this is the whole LAL test mechanism. Now let's quickly see the material required. Before understanding the material required, let's quickly see the flow chart that is showing the LAL test mechanism. So as you can see this figure, so there is an outline of coagulation of endotoxins, that and beta 1 3-glucane in Limulus lysate. So this Limulus lysate, they contain a specific recognition protein for bacterial endotoxin, that is factor C. So what is factor C? So factor C, it is considered as a you can say this is a Limulus lysate. They contain a specific recognition protein. So factor C, it is a specific recognition protein for bacterial endotoxins. Okay, now you can see factor there is another factor, that is factor B, or you can say factor G. So this factor G, it is a beta 1 3-glucane, and they are binding of endotoxins, or you can say beta 1 3-glucane to these is a protein activates the coagulation of the lysate, and they produce a clot, as you can see. Okay, so what they are doing, they are producing a clot. So the coagulation include various there are several specific proteinases, as you can say, as you can see, showing very restricted substrate specificity, The quantitative determination of a clinical material, or you can say synthetic materials, are applied, that you can see in this whole flow chart. So I'll post another video on a complete every step on this flow chart that shows the LAL test mechanism. Then what are the materials that are required? So the first material is the food sample. Then you required a clean and a dry test tube. They required a Limulus Lysate reagent, then a water bath incubator 37 degrees centigrade, then a wax marking pencil, then you need a sterile 0.1 and 1 ml tip autopeptide. So what is a procedure? So prepare the serial decimal dilution of sample using a normal saline. So you have to prepare a serial decimal dilution of a sample using a normal saline. Then you have to take a equal volume of diluted sample, means here the volume should be equal of the diluted sample, and a Limulus Lysate reagent in a clean and dry test tube, and mix the content gently. So what are the things that are required? So you can take a equal volume of diluted sample and a Limulus Lysate reagent in a very clean and a dry test tube, and you have to mix it very gently, means the volume of both the diluted sample as well as the Limulus Lysate reagent should be equal. Okay, and you have to mix it very gently. Then you have to put the test tube in the incubator, or you can say incubate the test tube in the water bath. And what should be the incubator temperature? Means 37 degrees centigrade, and for 4 hours. So you have to incubate the test tube in the water bath incubator, and you have to keep it at the temperature of 37 degrees centigrade for 4 hours. Then after incubation is over, after the 4 hours incubation is over, then the test tube should be inverted, means that you have to invert the test tube. And after inverting the test tube, you have to see the flow behavior, means how the liquid is flowing in the test tube. So you have to clearly see the flow of the liquid, means after incubation is over. Now what you have to do, you have to take the test tube and you have to invert the test tube. And after inverting the test tube, what you have to visualize or see the flow behavior of the fluid in the test tube. Now if the mixture remains unchanged, means there is no change in the mixture, means the mixture remaining unchanged, the run down the test tube wall indicates the negative LAL test, and thus endotoxins are absent in the sample dilution. So this point is very much important, that if the mixture remain unchanged, means there is no change in the mixture, and run down, and what you have to do further, you have to run down the test tube wall. And so after that, if the mixture remaining unchanged and it is run down the test tube wall, so what does it indicate? So it indicates the negative LAL test. So what they are indicating? A negative LAL test, and thus endotoxins are absent in the sample dilution. So this clearly indicates that there is no endotoxins in the sample dilution. But if the firm and the opaque gel is formed, means if the gel is firm and opaque gel, if it is formed, and it stick to the bottom of the test tube, and if it is sticking to the bottom of test tube when you are inverting the test tube, so if there is they stick to the bottom of the tube, so what does they indicate? So they indicates the positive LAL test, and thus endotoxins are present in the sample dilution. Clear? So when it is not stacking to the bottom, so it indicates that there is no endotoxins means if it is running down the test tube, so there is no endotoxins are present. But if it is stick to the bottom means if it is sticking to the bottom, so what does they indicate? So they indicate a positive LAL test, and it clearly shows that endotoxins are present in the sample dilution. So this is the procedure. Now moving further, so after that you have to prepare a observation table like this, in which you have to put one sample dilution opaque gel formed, and you have to interpret the result. So you can perform this experiment in your respective labs, and you can observe the readings, and you have to make a table like this, and you have to further uh, interpret the further results. So for the result and interpretation, of course the students, you have to perform these experiments in your respective labs. So that brings to the end of the lecture. This is the blank screen where I'm just writing some important questions, so you must be aware of the most frequently asked questions. So the first question in this type of experiment, whenever you are going to give any viva or any interview, so the first question is, what is endotoxin testing? So the first question is always there in interview or in examination, that what is a endotoxin testing? So if you see, this answer for this is endotoxin testing, or you can say bacterial endotoxin test. It is a in vitro assay that it is of course performing in the laboratory and used to detect the bacterial endotoxin. So they can be accomplished through several methods such as Limulus Amoebocyte Lysate, that is LAL, that we have just studied. There are other techniques also, like lysate in blood cell from a horseshoe crab, to detect the presence of bacterial contamination in the product. Or there are recombinant cascade reagent RCR. It is another type of a method. And of course, the most frequently used is a LAL, that is Limulus Amoebocyte Lysate. So what is endotoxin testing is? So endotoxin testing, it is a type of a bacterial endotoxin test, is an in vitro assay that is used to detect the bacterial endotoxins, and they can be accomplished through several methods such as Limulus Amoebocyte Lysate, that is LAL, RCR. RCR, it is a recombinant cascade reagent. So this is the definition or the answer for this. Now the most frequently question, second question is, what products need to be tested for endotoxins? Again, this is very important, means what are the products? So what are the products? So all injectable pharmaceutical, I'm again writing the question so you can note it down somewhere. So the second question is, what is what product need to be tested for endotoxin? So the answer for this is all the injectable pharmaceutical products, cell and gene therapy products, the medical devices, or you can say the dialysis product. Even some of the food products can also be used, are the products that that is to be that is there to be tested for endotoxins. So the next or the most frequently asked question that is the second is, what is a rapid progen test, that is RPT. So this question you have to google it, the answer for this, and in the next lecture we will be going to discuss this thing, that what is a rabbit rabbit pyrogen test. Okay, so these are some frequently asked questions, so you should be aware of all these questions. So that brings to the end of the lecture. If you have any doubt, any query related to the lecture, you can simply leave your queries in the comment section below. As far as the lecture is concerned, what all we have covered in this lecture, we have seen the introduction part, and after that we have seen the experiment, that is detection of endotoxins in food sample by Limulus Amoebocyte Lysate test. This is the theory that all we have discussed. We have also seen the difference between the endotoxins and exotoxins. We have seen this flow chart, so I'll be making a separate video on this LAL test mechanism. We will going to um, discuss this LAL test mechanism in detail, and we have seen the material required to perform this test. This is the procedure, that how this detection take place, and this is the observation and result. So for more understanding, students, you have to perform this experiment in your respective laboratories, and at the end we have seen some important 3 questions that is always been going to be asked in your interviews, in your viva, so you must be aware of all these 3 questions. So thank you, students, for watching. Please do like, share and subscribe, and if you have any queries, anything related to the lecture or related to any course, you can simply visit our website, that is www.itlsacademy.com, and this is our helpline number, that is 7080833450. You can simply call on this number and get updated, or if you want to enroll in any courses related to the life sciences, related to pharma, related to any other like computer courses, so there are various certificate courses that we are offering at a discounted prices, so you can simply call on this number and get updated, or you can simply leave your queries or anything on this number. This number is also there in the WhatsApp, so you can simply leave your queries, anything you wanted to ask, on this helpline number. So we are always there to help you. Thank you again. Thank you for watching. Yes, don't forget to press the bell icon to get more update. Thank you.